Hello everyone, my name is Jenifer Camila. I'm a postdoc researcher at the University of Illinois, working under the supervision of Dr. Alexander Lipka and Dr. Martin Ball. And today I'm going to talk about how you can use gene expression to improve genomic selection models in maize breeding. As many of you know, the global food system faces serious challenges, including climate change, land degradation, and population growth. Because of this, we need faster and smarter ways to develop better crops, and advanced plant breeding technologies like genomic selection can help address this challenge. Genomic selection improves productivity and supports food security by using thousands of DNA markers to predict the performance of plants that have not yet been tested in the field. This allows breeders to save time, reduce costs, and increase genetic gains, especially when traits are difficult or expensive to measure, or when testing across multiple environments is needed. In maize breeding, for example, genomic selection is especially useful for predicting the performance of new hybrids because it's not practical to test all possible crosses on the field. However, traditional genomic selection modules only use SNP markers, and this type of markers are static. They don't capture how plants respond to changing environments, like, for example, heat or cold. So that's a limitation. To address this, we are working to include gene expression and gene co-expression data in genomic selection modules. Gene expression tells us which genes are active when a plant experiences stress. This helps us understand how plants respond to their environment. That's why this information can be added to genomic models to make them more powerful and accurate. So, the main goal of our work is to incorporate gene co-expression information into genomic selection models to improve hybrid prediction across different environments. For this, we grow maize seedlings under control, heat, and cold conditions. Then, we collect gene expression data from both leaves and roots. After that, we build co-expression networks, which show how genes interact. Our next step is to include this data in genomic selection models and compare the results with models that only use SNP markers. We expect this work to generate valuable insights. It will help us understand how plants respond to the environment at the molecular level. And with this knowledge, we can build better prediction models. That means more accurate selection and maize hybrids that are better prepared to face climate challenge. Thank you very much for your attention. If you have any questions or ideas, I would be happy to hear them. If you can also reach me by email or LinkedIn. Bye bye.